In DNA extraction, we will use ATL buffer, proteinase K, AL buffer, 100% ethanol, AW1 buffer, AW2 buffer, AE buffer, microcentrifuge tubes, spin column tubes, a centrifuge, both 10 to 100 microliter and 100 to 1000 microliter pipettes, a vortexer, and a hot water bath. To begin, take your specimen, or if they are large, a leg of your specimen, and put it in a brand new 2 milliliter microcentrifuge tube. Add 180 microliters of ATL buffer to the tube. Add 20 microliters of proteinase K to the tube. Vortex the tube with your specimen in it for 15 seconds. Make sure your specimen is in the solution. Tap the specimen back into the solution if it's out of the solution. Place the tube specimen into a floater and then into the water bath. Let it incubate at around 56 degrees Celsius for 6 to 8 hours. While the specimen is incubating, label a microcentrifuge tube and a spin column tube with the specimen's ID. Now that 6 to 8 hours has passed, vortex the tube with the specimen in it for 3 seconds and add 200 microliters of AL buffer. Vortex again for 15 seconds. Add 200 microliters of 100% ethanol to the tube and vortex until the white precipitant disappears completely. Pipette the liquid contents into the labeled spin column tube and centrifuge for 1 minute at 8,000 RPM. Make sure to balance your samples when centrifuging. Remove the specimen from the tube and place it back into its original tube to be stored. If a leg of the specimen was used, toss it. Pipette the liquid in the bottom column back into the top column of the tube and re-centrifuge. Dispose of the collection tube and place the filter tube in a new collection tube. Add 500 microliters of AW1 buffer and centrifuge for 1 minute at 8,000 RPM. Dispose of the old collection tube and place the filter in a new collection tube, followed by adding 500 microliters of AW2 buffer and centrifuging it for 3 minutes at 14,000 RPM. Dispose of the collection tube and place the filter tube into the previously labeled microcentrifuge tube. Add 75 microliters of AE buffer and let it sit at room temperature for 1 minute. Then centrifuge it for 1 minute at 8,000 RPM. Remove and dispose of the filter tube. Keep the newly collected contents. This is your extracted DNA.